Welcome to Study Loop.
Here for this question, we're looking at the different modifications that can occur to our histone proteins. There are two different types of modifications that we're going to consider, which will be the methylated form and the acetylated form. So the function, first of all, of histone proteins is to bind DNA and to help it condense inside of the nucleus. So as I mentioned, there are two forms. There's the methylated state, where your histone proteins are going to be held close together, and the acetylated state, where they will be much further apart with less interaction. So in each of these, you're going to have DNA bound and coiled around those histone proteins. In the methylated form, you're going to have the DNA very closely bound and tightly wrapped around the histone proteins. Whereas in the acetylated form, it's much looser and you have lots more open areas of the DNA. And this is important because inside those open areas, you're going to have those genes that need to be transcribed and translated. And when they are in this open state, there's room for the polymerases to fit in and conduct their processes to create those mRNAs. So acetylated histones allow for the increased transcription of these genes. Whereas in the methylated form, these genes are sort of hidden away and they're closely packed so that the polymerases cannot reach into that area and transcribe and translate the genes. So the methylated form of histone proteins have a decreased rate of transcription. So here in this scenario, when we're talking about a silent gene, it's going to be in this methylated form because polymerases are not able to read and transcribe the gene due to the closely packed together histone proteins. So the drugs affecting this are going to convert this methylated form of the histones into the acetylated form, where these genes are open and able to be processed. So here the best answer choice for this question is going to be A, that these drugs are going to prevent the methylation and cause the acetylation of the histones instead.